messages.
So just to quickly, so what I meant, this was mentioned, then the academic innovation, regulatory innovation, global impact, and the dedicated clinical infection unit that is called INFECTA. INFECTA is there to accelerate pharmaceutical development in infectious diseases through innovative research methodologies, strategic collaborations, with a commitment to improve global health outcomes. So 100% of the shares of INFECTA are owned by the foundation. So that means that we actually work as a non-for-profit purpose. And one of the directors is Ingrid, who is going to present after me, and the other one is Meta Rustenberg, who is a professor at the LUMC, the Leiden Medical University. We're going to build a building with 20 beds on BSL-3 level to do the clinical research, an outpatient clinic, and a lab facility on BSL-3 GMP to produce the viral strains or the pathogens that we need for the controlled human infection models. We have a very strong legacy from the Center for Human Drug Research and the Leiden Medical University. The Center for Human Drug Research is a full-service CRO that already exists for 37 years, and that's really where our operational excellence comes from. And the Leiden Medical University is where our scientific excellence is coming from. So what is it actually that INFECTA does? It's a full-service early-phase CRO, regulatory compliant drug research, the development of novel clinical research methodologies like biomarkers and controlled human infection models. Ingrid will tell more about what are controlled human infection models and why are they working so well for infectious disease research. We can develop vaccines, therapeutics, antivirals, biologics, but we also can help with early phase clinical drug development support and consulting, because often we also see that the translation from preclinical to clinical not always works optimal, and we have a lot of experience in that as well. This is an overview of all the models that we have in the Netherlands. So it goes from viral models like dengue and influenza, bacterial ones from C. diff, pneumococcal, but also parasites like hookworm and malaria. And we stand for high quality, innovation, and being connected. And if there's any questions on controlled human infection models or these clinical trials, feel free to reach out. And then I would love to hand over to Ingrid for the rest of the story.
Thank you, Jeroen. Before I start, can everybody hear me? Because there was an echo. Yeah, you can hear me all?
Good. Yes.
Perfect. Okay. So I can continue then. I'm not so familiar with Zoom, but I will try to share. Wait and share my slides and put them in presenter mode.
Perfect.
Can you see it in presenter mode? Okay, you see my slides?
No, just the slide deck right now.
Okay, so I think I have the same problem as you, or is it now better?
Oh, yes. Yes.
Perfect. So I'm very happy that I managed this. So the rest is to give the talk to see what we do at INFECTA. Before I start about our clinical work and about our research, I just want to introduce myself very quickly. My name is Ingrid de Visser-Kamerling. I'm working as a research director at the Center for Human Drug Research in Leiden, the Netherlands. I'm supervising all the clinical trials on vaccines and therapeutics in the field of infectious diseases at CHCR. And next to that, I am one of the directors of INFECTA, what already was told by Jeroen. Before I start to explain what we do at INFECTA, first I want to show you something about clinical drug development in general, but specifically also for infectious diseases. The thing is that when you start with the clinical trials, you first start in healthy volunteers. And what you normally do traditionally is to see whether your compound is safe, is well tolerated, and you will also look at a certain dose. So most of the time it's dose escalation, single dose escalation study. And when it seems that everything is safe and well tolerated by the healthy volunteers, you go into the phase 2. Phase 2 is mostly very, very small trials already in a subpopulation of patients. And in the phase 3, that is when the efficacy is really investigated of the compound. So that is a very moment of truth. Does my compound do what it expects to do? So it is really the trial in patients to see whether it works or not. And what you normally see is that most of the time it doesn't work. And what you see here is that at the y-axis is the number of candidates and the development phase is the x-axis. So that you see that the bar on phase 3 is significantly lower than in the phase 2 and the phase 1. That is because it really is the moment of truth, though, at that moment. The thing is also that the further you get in your developmental phase of your clinical compound, that it will become more expensive. So you can imagine if you want to see whether your drug works or not, or your vaccine, you really need a large population of patients. That is very expensive. So the red line is the line that shows the expenses, so the price of clinical trials. So you see that the more expenses are done at the later phases of the development. That's not what we want. It says you rather fail fast, of course, but you really want to succeed fast at all as well. So the thing is that we really try to, at INFECTA and also at CHDR, to see if we can put that moment of truth. Does your compound work or not? What you normally do in patients, that we try to move that a little bit forward. So already in your phase 1. The thing is that you then need some models or biomarkers that show efficacy in healthy volunteers. So it's not real patients, but it's tried to mimic a patient situation in a healthy volunteer. That increased the cost a little, but if your compound shows efficacy over there, you have more confidence that it will also work in patients. So it gives you more confidence to move on to the next stages of the clinical development. And if you have like these positive results, you can also more easily find venture capitalists to invest in your compound because it's very promising. So there will be a little bit more expenses at the beginning, but it gives you confidence that your compound is working, or maybe it's not working at all, but then you rather know it very soon in your clinical development. So that is our philosophy. I cannot see you, but maybe you're thinking, but healthy volunteers are not patients, and how can you find biomarkers to see whether a compound or a vaccine is working in a non-patient, so in a healthy volunteer? So we have a legacy of developing biomarkers and models at the Center for Human Drug Research to see whether we can put a healthy volunteer into a state of disease. We can do that at dermatology research or pain research or internal medicine. We all have different models. For example, I can explain the internal medicine model we have. That is a glucagon clamping. What we do is we clamp glucagon. That means that the glucose will be on a certain level, a clamp level, so at a certain higher level than normal. And at that moment, we can regard the healthy volunteer as a diabetic patient, and we can see if a diabetic compound treatment is working or not. It's not a real situation, but it gives you some sign of evidence whether it's working or not. So we can call it a biomarker. We also have the LPS challenge. LPS is the outer shell of bacteria. When you put that in a subject intravenously, what you will see is that the patient will, just simple, it's all kind of graphs, simple, you put them in a state of inflammation. And we have seen and investigated that this model can also be of value when you are developing and seeing whether a anti-rheumatoid arthritis compound is working or not. So we always try to see what is our client wanting us to show their compound, and then we can develop the model next to it. So for infectious diseases, it is we developed the controlled human infection models. what we do in a controlled human setting, we will inoculate or infect a healthy volunteer. So that these models are already existing for 10 decades. And it's not only to see the interaction of the immune responses to the infection or to see how the pathogen works in the healthy volunteer, but also to evaluate the efficacy of vaccines and drugs. That is what we do. So we try to develop models that we put a healthy volunteer in an infectious disease state and then see whether a compound or a treatment or a vaccine works or not. At the LUMC, together with CHDR, we did a very nice and good trial on a very nice compound of Merck. This was an anti-malaria drug. What we did, what we can do is when we infect people with malaria to see whether the drug works or not, what we can do, we can put, as you see on the right, we can put these sporozoids into a vein, and then the healthy volunteers will be infected by malaria. Or we can infect the healthy volunteer with malaria by mosquito bites. In this case, at the malaria study for Merck, we did the intravenous one. What you can see here is the different types of how we can use the model, the controlled human infection model for malaria. We can test a vaccine, and we can also see whether medicines work. What you do is when you want to see whether a vaccine works or not, you give the vaccine to healthy volunteers, and sometimes you wait for a month or a couple of weeks, and then you infect people. If you still can infect people, your vaccine was not working, and if it's not possible, you have a very good vaccine in hand. You can also see whether you can investigate treatments, so antiviral or anti-malaria drug, whether they can decrease the symptoms, and then you do it the other way around. So you first give the infection, you wait for symptoms, and at that moment you can see whether your compound works or not. So these are the two types, or this one model, but we can use it for two types of new treatments. The study design was as follows. The sporozoids were, the shelf life, so the inducibility was very short. So what we did with the healthy volunteers, we inoculated, so we infected them at the Leiden University Medical Center, which is located only 5 minutes walk from CHDR, and we inoculate them with the malaria sporozoids at the LMC. We walked back to the CHDR, and we gave them the compound, which was a solution. So it was a capsule that we had to open into water, and the subject had to drink it. And this model can be run outpatiently. So after the drug intake, the subject were discharged the day after. And every day they came back to the CHDR for a qPCR. qPCR is a very sensitive method to detect the sporozoids in the blood. For malaria, malaria is a disease that the sporozoids first go to the liver, stay there for at least a week, 7, 8, 9, sometimes 10 or 11 days, and then the sporozoids move into the blood, and we call it the blood stage, and that is the stage where subjects get the symptoms. For this model, at the moment we had a positive PCR, we cut the whole study by giving a rescue medication to the subjects, and sometimes we had to stop, or we stopped the infection already before subject had symptoms, because sometimes the qPCR is so sensitive that you already can see that there is that sporozoids are present in the blood, but they still don't have symptoms. So it is not a model that subjects get very ill and get a lot of malaria symptoms. The symptoms we saw was mostly a little bit chilly, headache. There was like the most intense symptoms we saw. So yeah, and that is also very good to show you here. So normally what you do is you put your compound into the best preclinical model. Sometimes it is dogs, sometimes it is like pigs. Most of the time for respiratory viruses it's like ferrets and hamsters. And then you do a pharmacokinetic prediction and a PK/PD modeling, so pharmacokinetic and pharmacodynamic. So you really try to model what dose would be effective of your compound in a healthy volunteer. So I was also involved, and we did a lot of calculations, and we were like 200 milligram, that will be the lowest dose predicted from the preclinical models that will be probably already have some effect. What we saw at 200 milligram, we saw already that it was working. This kind of models can be done in very small groups. It was only 4 subjects, 1 on placebo and 3 on the Merck compound. What we saw after 11 or 12 days that only 1, it turned out to be the placebo, got symptoms or had a positive qPCR. So for this one, it was not a dose escalation study, but it was a dose de-escalation study. So we started with 200. We saw complete 100% protection. Also with 100 milligram, we saw 100% protection. So we decreased up until 60 milligram. No, 30 milligram. And so that is not normally the models are like more predictive. Here it was like, no, it was not predictive for the protection against parasitemia. It was very good for Merck because they saw already in their very early start of the clinical trials that our compound was working against malaria. So this gave Merck more confidence to move to the next phase, the clinical trials. What you can also do is develop models for other diseases, such as respiratory viruses. This is not a model that we already have in hand, but we are working on it. It is a controlled human infection model for influenza. This is work from Hayden and all more than 25 years ago. What they showed is that when you infect a person or persons with influenza, what you can see is that there will be symptoms and there will also like viral load, and that if you give antiviral drugs such as oseltamivir, that both the infection goes down or the viral load goes down much quicker, and also the symptoms are not as prominent as when you don't give the drug. So what we now are in, we are in Fluniversal. Fluniversal is a European program, a lot of partners from all over Europe, and the aim is to develop a vaccine that will work against all influenza types. So we call it a universal flu vaccine. What we do is that we will use a controlled human infection model for influenza to show the effect of the universal vaccine in that consortium. Yeah, and then just a small, because I think I'm running out of time, I move on a little bit quicker. We developed a rhinovirus challenge in Leiden. There was a company in Vienna. They had a compound against common cold. Rhinovirus is common cold. So what we did, we infected the subject with rhinovirus common cold, and we gave also the treatment. The thing was that treatment was not working. So we were thinking that the client was very disappointed. And of course, they are disappointed that the drug is not working, but it was the very first clinical trial. And they already now knew that drug was not working because we were able to infect almost all subjects, and all subjects had symptoms. So the drug was not working, and it was not working. So you rather fail fast than that you continue with a compound that does not effective at all. Yes, this is a little bit of advertisement, I don't know. It's like what we all do. We do rhinovirus, malaria, schistosomiasis. We have the largest global chimps in the Leiden area. At this moment, at this time, we are currently infecting subject with RSVs or respiratory syncytial virus to see if we can get symptoms. This is in a very, very big consortium, also European consortium, called InnovaVac. There's big pharmaceutical companies involved, such as Sanofi. They really want to have a more recent RSV challenge model. So we are now trying to see if we can have that model enhanced too. So this is really what we think that will work. So that we think when you have a RSV compound or a vaccine, that in this model, you can get a answer whether it's your compound is effective or not, is can be shown very early in the clinical trial. Just some pictures of our clinical unit. This is the 7th floor of the CHTR building, where we have at this moment 8 isolation rooms. These isolation rooms are applicable for viruses and or pathogens up until class 2 pathogens. And this is not sufficient for pandemic viruses, pandemic pathogens. So that is why we are at this moment trying to build a new building in which we have 20 isolation rooms with beds under BSL-3 conditions. So that was my presentation. So that were just backup slides about INFECTA and INFECTA, but I was really happy that Jeroen already presented you INFECTA and INFECTA.